Before we start, let me introduce our speaker today, Romain Hollands. Romain has a background in human neuropsychology. He studied at Maastricht University and he specialized in cognitive and clinical neuroscience in drug development and neurohealth. He worked on autism models of rodents at Hoffmann La Roche in Basel. And afterwards, he worked as a teacher at Maastricht University at the Faculty of Psychology and Neuroscience. And now he's an account manager at Noldus and scientific consultant. So that's the background of Romain. We're going to start now. So Romain, the floor is yours.
Thank you, Lianne, for the introduction. So I'm Romain Hollands. I want to add to the introduction of Lianne that how I came to Noldus is actually because I worked with Noldus products myself during my research. So when I was working at Hoffmann La Roche in Basel, I worked with E2Vision XT, the Phenotyper, and the Observer XT. I used this equipment and the software for the study of autism models in mice and rats. That is my introduction to Noldus, and I was. It's true if I say that I was so enthusiastic about the products that is what actually connected me to the company. So now I am working for Noldus over a year now as an account manager mainly. And today I will be talking about the phenotyping of your rodents and the mapping of short and long-term behavior, and specifically the switch that we see from short to long-term measuring in rodents and why that is important. So first of all, the history of measuring behavior can be summarized with 3 items, which is a pen, a notebook, and a stopwatch. These are 3 items that most of you are probably aware of, and most of you still use sometimes if it's necessary. And when we have these 3 things, we need 3 other things, which are spontaneous behavior or induced behavior, and we need rodents. So these are the 3 things necessary. And if we then look at the behavior of animals, there are also certain criteria that we want to make sure are matched to be able to do our research. And that is that the criteria of the behavior to be measured is, first of all, the behavior should be observable, recognizable, because that is always, when we look at an animal, it should be observable. It should be short, because we have a limit of manual scoring, how it always used to be. It should be specific. We need to pinpoint that behavior. And it should be stable, so that means not being incidental. It should be a behavior that within a certain category happens continuously. Should have a relative quick onset, and again, this is the limitation that we mostly have with manual scoring. It should occur in validated paradigms. Is of course very important. So, with that basic, and with the fact that we always needed to manually score, and we always needed to live up to these criteria for measuring the behavior in our rodents, we came up with certain experimental paradigms. And I want to tell you, please don't look at all the specific details mentioned in the following tables. It is just an overview of how many experimental paradigms there are. So first of all, we have basic motor and sensory functioning paradigms. We have learning and memory paradigms. We have social behavior paradigms. We have paradigms regarding anxiety and depression-like behaviors. And we have paradigms regarding reward. So this is a section of it. Of course, there's more than only this. So following these criteria, we have these paradigms, and I'm sure that all of you recognize at least most of these paradigms that we see here. So if we zoom into how research apply these paradigms, we find the following. So everything now in the black box is our paradigms that are 99% of the time we conduct them in the short term. And why we do that is because the paradigm is generally a paradigm that we only can conduct in the short term. Give and take maybe some exclusions if we change the paradigm a little bit. And now in the red boxed paradigms, these are paradigms that we can apply short and long term, but generally researchers apply them in the short term. And keep this in mind, because this is something we will get back to. And the reason why researchers apply these paradigms mostly in the short term is, again, the limiting factor of time and manually observing the animal and manually be there in order to make sure that all the data is gathered. So how do we come to these paradigms? Well, there's a golden rule of validity, a golden rule of validity that we need to apply in behavioral research if we want to translate the information from an animal model to a human model, which is logical. And generally speaking, this is the face validity that we look at. And the face validity then here says is that the symptoms of the animal model need to mimic the symptoms of the human condition. And this, of course, we also see if we look at the paradigm. So if we in humans conduct questionnaires, then in animals, we also like to do something in regards to scales that we can put into there. Example, Grimace scales. Cognitive tests in humans also relates to cognitive tests in animals. For instance, the novel object recognition, choice paradigms, acute physiological measurements in humans, we also measure acutely then in animals. So for example, EEG, heart rate, weight, grip strength. And then longitudinal acute physiological measurements works the same, but then at different age points. And then public data health available in the databases for humans is now also, of course, with the development of internet, publicly available metadata of comparable studies. So all in all, human and animal research is quite in relation with that. We really look at the face validity and it seems to match. However, there seems to be a definite shift in importance from short to long-term observations in humans. Why that is, I will follow up to later, but let's critically look now at the short-term observations in animals, because there's a few things to consider here. So in animals, if we do short-term testing, we know that the tests that we conduct and the paradigms that we use, they are robust if we make sure that the experimental conditions are equal every single time. Short-term behavior is validated if we make sure that the paradigm is applied correctly. And short-term behavior is very practical to work with, of course, because it is in the short term. And short-term behavior is easy to understand. At least we think that it's easy to understand. However, it is still depending on the human interpretation of an animal's cognitive behavioral domain. And that's a complicated sentence, so read it again if you missed it the first time. It's depending on the human interpretation of an animal's cognitive behavioral domain. So we decide as humans what is happening inside of the animal, which is fine because sometimes it makes a lot of sense. However, we will also have a subjective scoring factor. A subjective scoring factor of giving our own opinion about certain things that are happening, and that might be different from the colleague of yours or another lab or another university or another city or another country. Everyone has the subjective scoring factor. This can be reduced by use of, for instance, automatic tracking software, such as e2vision. So if we track an animal from place to place and we track it using software, the software will track it every time the same way. And lastly, it's good to mention is that we all know that neuroscientific research or anyway, animal research is struggling with replication. We see this every single time. Yearly, there's so many papers published that say we didn't find the same results as another researcher found. And there's reasons for it. And sometimes it's the way the research is conducted, but in general, often it has to do with the subjective scoring factor that we have and the paradigms that are not strictly followed. So saying this, should we get rid of short-term testing? Well, no, absolutely not. We should not. Some examples, acute ischemia. We want to study acutely. It doesn't make sense if we study it for a very, very long time if we're just interested in the acute effects of it. Acute stress, we need to measure that acutely. Again, if we are interested in the long-term effects, we can study that long-term, but acute stress, we measure acutely. And short-term memory, for instance. Short-term memory can be minutes, can be hours, can be days. However, short-term memory, we need to measure in the short term. Again, it doesn't make sense to measure short-term memory over months of time. Maybe the development of it, but not the short-term memory per se. However, we should be very cautious with short-term testing because external factors can have a large impact on short measurements. And now I will explain what the reasons are. So that's the stress on laboratory animals. These graphs are exaggerated, but they come from real research that has been done. The literature you can find at the bottom. Short-term stress is, for example, handling, transport, and procedures done with animals. And then the lines depict the following. The blue line depicts the normal behavior, and the red line depicts the stress behavior. And on the y-axis, we see the percentage of expression. So if the blue line goes up, that means that the normal behavior is more expressed than the stress behavior. And if the red line goes down, that means that the stress behavior also goes down. And on the x-axis, we see the time in minutes. And what we generally know from testing animals in the short term is that when we handle our animals and when we place it in an environment, the first behavior that will be expressed by the animal will be stressed behavior. And after some time, that stress behavior will go down, the influence of stress will decrease, and the amount of normal behavior will go up. So that is really good to consider when doing short-term testing. On the other hand, we have long-term stress. And long-term stress has to do with captivity, noise, restraint, the general environment in which an animal is being held. And we do our best to keep our animals in a good environment, but sometimes it's not so easy, especially considering that it's never the natural environment of the animal. And again here, x-axis, y-axis quite the same. However, now we're speaking in time of weeks instead of time in minutes. And we see that stress behavior after long term of captivity goes a little bit up, and the normal behavior reduces a little bit. So we need to find a trade-off within that. We need to make sure that when studying short-term behavior, it is not stained with stress, and when measuring long-term behavior, that it's depicting normal behavior. So how to be cautious with short-term testing? Well, first of all, you apply a standardized habituation phase. If you take your animals up to your testing room, you make sure that you have some time to habituate to the environment. Consider carryover effects after multiple tests. If you tested the animal 10 minutes ago, an hour ago, you are very certain that you will find some effects of that previous test on the current test that you're conducting. Make an adequate study design. As you, as experienced researchers, you know how that works. And then also very importantly, consider the assessment schedule and the influence of dark light. So the graph to the right is a little bit darker now. It will increase in the following slide. This has to do that we know that rodents are nocturnal animals. And most of their behavior, they will naturally express during the dark phase, not during the light phase. Also in the morning, they will depict other behavior than in the afternoon. It's the same with humans. Standardize your metrics, standardize your markers. Make sure you measure the same way every time and analyze your data appropriately. This is statistics 101 as I put it here. Make sure that this happens. And if you're talking about the standardization of your metrics and markers and analyzation of your data to be happening in a proper way, and definitely also as considering the assessment schedule and the influence of dark light, then E2Vision XT, the automated tracking software, can help you with that. And most of you are probably aware of E2Vision XT and the tracking software, but just as a quick reminder on it, E2Vision XT automatically detects your rodent. So it's not up to you that you have to track your rodent throughout the cage. It tracks the speed, movement, location, behavior of your rodent 24/7 if you wanted to. There's no manual scoring needed, but you can if you want, but it's not needed. It has quick integrated statistics and graphing. So the data and the view of what happened is all visible at the same time. And you can plan your experimental protocols in the software. And how does this help you? Well, considering the assessment schedule and the influence of dark light, you only have probably 8 hours available in a working day. Now, I know with researchers that is sometimes way more than 8 hours. If you have a software that tracks your animal and you don't have to be there physically, there are so many more animals that you can test at the same time probably, so many more studies that you can do, and you have so many more chance that more animals are tested within the same construct of time that you want to test. Plus, you have the assessment of schedule of dark light because you can test them 24/7. You don't need to necessarily be there. Standardizing your metrics markers has of course to do that. E2Vision XT measures the same way every single time, and analyze your data appropriately means that all the data that is being captured is synchronized nicely with each other with a view of what happened. So I have a nice practical scenario of how I always like to compare the short-term behavior that we look at in animals. This is always my opinion, how I viewed it when I was doing research, and buying a new car. So, or a second-hand car, works for both. You go to a dealer and you look around for the car that fits your needs. Well, in short-term, in animal testing, that is the disease model and the animal model that you picked out that you want to investigate. You ask the dealer about the details of the car, which is, for instance, previous research you've done on that animal model. You look at the outside for any damage and the aesthetics. So is my animal healthy according to the expectations that I have? And you take the car for a test drive. And in our case, that would be short-term behavioral testing of my animal. And at that moment, you decide whether to buy or not. And that is the experimental results that you have. That is the conclusion of it all. So after that moment, after that visit to the dealer and that test drive, what do you know? Well, you know probably how the car performs during a short drive. For example, acceleration, start-stop, stability on the basic known road. What about the long term? How does the car handle a long distance? What happens if I brake 100 times? Is it stable during rain or snow? And maybe does my partner like it, but that's maybe not too important now. But you don't care about that too much. Because why don't you care about that too much at that moment? Because you've got warranty. However, it still means that short-term testing gives you short-term effects. And with our rodents, we don't have that warranty. If we put it back in the cage, if we did that short drive, we don't know how it performs in the long run because we're not looking at the long run. So we need to include the long run. We need to include long-term testing. So let's have a quick look at the history of the long-term measures. So the measures of human have developed from focusing on a broad spectrum and having a few instances of data collection to using smart devices, mobile devices, and having the focus on personalized medicine. We want to map a single human being as well as possible. And following that road, this has led to the development of digital biomarkers and also yearly digital biomarker summits in which we discuss digital biomarkers. And what is digital biomarkers? Well, Roche, Hoffman La Roche, it's a little bit biased because I work there, but they call it the measure of you. So what are these digital biomarkers? Digital biomarkers are measures of small changes over time. They are previously undetectable, unmeasurable, because they are so small at a given moment, but they give insight to new symptomatic domains. And from that, we can develop new digital endpoints that allow more successful and rapid clinical development because it's much more detailed and it's much more stable in that sense. So for instance, in a human, that would be sleep, looking at sleep efficiency, total awake time, daytime naps, general activities, circadian rhythm, activities of daily living, the behavior that you do throughout the day. And for instance, the walking, the speed, the forward acceleration, and also the balance, fall, sway, and jerk, up and go. As you might notice, this is something that is very related to Parkinson research nowadays. And next to this data that we also have pharmacological data available. We have lab data available, wearable data, hospital data, survey data. It's a big collection of all the information that we have that give us an insight of really the disease and the behavior of a human during that period. So if you compare short to long, instead of having a spot estimate of the behavior, we measure the average behavior. And instead of having an indication, providing an indication, we have highly accurate results of what it actually all means. Instead of estimating the health impact, we can measure health impact. And instead of unwillingly measure measures of stress, we measure the natural behavior at that moment. Of course, keep in mind, sometimes you want to measure stress, but that is not what I'm speaking about at that second. But having this, what should we measure in rodents to get up to the same speed as we have in humans? So this is what we have in humans. Now, let's get the human away and let's put a nice mouse there. For instance, general activity would be the activities during the day, the behaviors of our rodent during the day, the activities, the on and off switches of certain behaviors, changes of activities over time, movement, speed of movement, accelerations, sleep, circadian rhythm, number of movements, the place of sleep, also sometimes very important. And for instance, the feeding behavior, time spent eating, time spent drinking. This gives us a good insight of the general behavior of our animal. So how to measure these long-term findings in humans and animals? How do we find the same thing as we have in humans? How do we find it in animals? And keep in mind, this is really important to make sure that what we do and what we test in animals is the same as what we test in humans. If we find positive results in animals, we need to make sure that under the same circumstances, we also will find positive results with interventions or medications in humans. And that is where it mostly goes wrong. We have positive results in animals, then you move to humans, and suddenly it doesn't work anymore. There's also, of course, other reasons, such as that our brains are not exactly the same, but leaving that behind, that is the basics of it. We need to translate our results. So from that, we need a measurement tool that is objective and doesn't need manual scoring. We need a suitable environment where the animal feels home, so we have natural behavior, and we need a subject of interest. So I have an example scenario here. Measurement tool that is objective and doesn't need manual scoring, E2Vision XT. Suitable environment where the animal feels home, Phenotyper automated home cage. Again, something I worked with myself. Explanation of what it is will follow. And we need a subject of interest. In my case, this was the Black 6 catnap to knockout mouse. A first introduction, what is the phenotyper? If we look at the basics of what the phenotyper is, I can summarize it in these points. It is a standardized testing environment suitable for mice and rats, right? Standardized testing environment. You can keep in your animals for 10 minutes or 2 weeks, because it's possible to, of course, have food and water present. It has a fully integrated camera and infrared light, so you can test your animal during day and during nighttime. It doesn't matter, it's constantly tracking the animals, and it has no influence of the daylight whatsoever. Works with the E2Vision XT software, and it has a proven track record, which I will show you later on. A little bit more detail, but I will just quickly go over it. You can do extensive behavioral mapping, including the feeding. You can use it for learning and reinforcement protocols by using light, sound, shocks, or pallet dispensers automated from E2Vision XT. You can map behavior and EEG data perfectly synchronized. You control your optogenetics automatically on the basis of the behavior, speed of location. You don't have to be present. You have a high throughput screening of the treatments that you give, many animals at the same time. You can do a circadian rhythm analysis, and you don't need to rely on sensors, but you actually see what your animal has been doing. So it's fully customizable. It has many walls that you can put in there. You put it to your taste, your specific research that you're doing. At the left bottom, you see a paradigm in which a pellet is being dispensed, and the animal walks to it, collects the pellet. Next to that, you see a picture of a setup that we have, a 16-cage setup. And at the right side, that is actually one of the phenotypers that I use. That's with the running wheel. But next to that, there's many people that use it. For instance, Roche, Boehringer Ingelheim, Orion, and Pfizer, they use the phenotypers, but also over 150 other users worldwide. So let's go to my setup, the setup that I used. I individually housed my animals. I had 3+ days available of observation. I had food and water available ad libitum. I had wild types, knockouts. I had a shelter present in the phenotype. I had a running wheel present. They were in a 12-hour cycle. We observed them, of course, with the infrared camera to make sure that also during the dark phase, we can see what it was doing. It was all analyzed with E2Vision. So let's have a look at the results. So first of all, this is the graph that you saw before, the percentage of behavior during the light phase and the dark phase. You see that the wheel rotations, that is something that more than 90% of the time that an animal is doing it, it's doing it during the dark phase, not during the light phase. And the light phase is the phase that we test our animals. Walking, rearing, drinking, eating, all automatically scored. This is behavior that happens during the dark phase. And then we look at grooming. That is again, behavior that's a little bit split because animals tend to do it throughout either darkness or lightness. And at the right, we see the number of running wheel rotations per day. And this was really interesting because this is where I found a new phenotype. When you put in your animal, so we took our catnap to knockout mice, put them in, and on the first day, we didn't see any difference between the wild types and the knockouts. So compare that to acute testing, no differences. But then on the second day, a pattern started to occur. And the third day and the fourth day, we saw that the wild types started to be very stable in their amount of rotations that they wanted to do in the running wheel. But the knockouts just increased in their behavior. They just run more and more and more and more. Results that we would have never found if we would have only tested them on the short term. Then the circadian activity pattern over 24 hours. Again, this one runs from 18 o'clock until 17 o'clock. Everything in the black dots, that is during the dark phase, and everything in the white is during the light phase. we saw a nice pattern. We saw some spikes, different activities during a different point of the day. And again, the behavior happens during the dark phase, not during the light phase. That's the phase where I was always testing my animals, but behavior happens during the dark phase. So I also investigated the sleep pattern and the stability, and I saw that the knockout mice, the autistic symptomatic mice, I need to call them, had more visits to the shelter during the light phase than did the wild types, which means that they were more disturbed. They woke up more often and they left and they walked, and just not one long sleep as the wild types had. They also didn't have a long sleep, but they had more stable sleeps. So comparing that, the expected versus the results that we found, I expected to find excessive grooming because that's what I found during the acute phase, but I found no excessive grooming in the long term. I expected to find excessive rearing, which I found during the short term, but I found no excessive rearing in the long term. I expected to find a disturbed activity pattern. And I did find a disturbed activity pattern. It was related to sleeping. I saw that the sleeping was a little bit more disturbed. And I expected to have more activity during dark than light. Not a result that I found during short term, but a general thought that everyone probably has. And of course, that happened too. And I found a new phenotype, which was the running wheel phenotype. So the long-term measurements led to new stable insights that are less subjective to short-term acute variances. And now keep in mind that graph with the behavior and the amount of stress and the influence of it. So this is the expected versus the results that we actually found. And then we move to the little bit of the conclusion of it all. So we have a common goal in animal research, and that is then to translate the results we find in animals to translate them to humans so that they fit well. And we have short-term and we have long-term behavior. And there's a few considerations that we need to take into account. We need to consider the animal behavior. An animal has a different circadian rhythm. An animal is quite impacted by stress, by handling and the environment. We should stay objective. Otherwise, we're not doing our research in the right way. You need to try, if it's possible, to take the animal for an extensive test drive. We can't rely on the warranty that we have with our car. We need to test the animal, really. We need to measure to know, but we also should know what you're measuring, because otherwise you're just doing something what you think is right, but you need to know what you're really measuring. You need to use the right tools for it. So in this case, that was the E2Vision XT automatic tracking software and the Phenotyper home cage. And I really want to stress, short-term measures are also important, really they are, but you need to apply them correctly. So make sure you have a standardized environment. So how to, for in this case, the long-term observations, make sure to use an objective automated software without manual scoring. So you have the same measurements every single time. Make sure, in this case, we have a fully automated home cage observation environment in which the animal feels comfortable. If it's always the location where it's been tested, if it can be tested for a longer time, the animal will perform more stable than if it's constantly in a different environment. And with that saying, if you're interested in that, I have a very nice paper available from a study that was done in Groningen, in Switzerland, and at Pfizer at a different location, and they were 3 different locations, and they had the same paradigm that they all tested in the same way with the Phenotyper cage, and they found the exact same data. They found the exact same results, and that is quite unique. To say that in these days that you find the same data at 3 different locations in the world with all different researchers, that is quite unique. That's a very nice paper, and happy to share it with you. So from that, I assume you have maybe some questions, and that's where Leon comes in again to guide us through and see what we can answer. Thank you very much.
Okay, Romain, thank you very much. Very interesting. So if you have any questions, please fill them in the Q&A. There's already one, I see, Romain. The question is that you said that you kept your animals for 2 weeks in the Phenotyper. Why not longer?
That's a good question. You can keep them in for longer. The thing to consider here is that the environment where you keep your animal in, I mean, they will defecate, right? They will maybe stain the environment a little bit. So you want to make sure that the environment that they are in is still clean up to the measures that you allow for the animal. So there are certain researchers that say we want to keep our animal in a clean environment constantly. But if you want to keep them longer and you're fine with that, please do so. And also, if you allow for switching off the bottom and switching off the insides of the cage to have a clean cage, that's also possible, but take into consideration you're then disturbing the animal throughout its standard behavior.
Yes, thank you. So far, there are no more questions, but let's wait a little bit. Okay, there's more coming. There's a question that says you showed data from mice in the phenotype. How about rats?
Yeah, rats is that's a good question. So how about keeping rats in the phenotyper? If we go back a little bit in the slides that I have, here we see actually a rat. Is that visible to you now? Yes, that is visible. This is a rat in the phenotyper, and this is running with the automated behavior recognition module on eToVision. And in this case, it's calculating the specific behaviors that the animal is doing, such as rearing, eating, drinking, grooming. So as you will now see, the animal will move forward and will start drinking, which is registered at the blue line that you see at the bottom data. And from that moment, it will also eventually move on to grooming. But what I want to say here is that, of course, you can keep rats in it. The difference is the size of the cage. The rat cages are a little bit bigger than the mouse cages. You can use both of them, but just to keep the environment friendly to the rats. So yeah, that's fine. That works nicely. I also did research with rats.
Mm-hmm. Then the next question is related. It says mice prefer company. Can you keep 2 or more animals in one cage?
Yeah, very good, relevant question. I did this both with mice and with rats. I tried this. What you need to consider in this case is the identity of the animal. Is it important to you to keep the identity of the animal? In general, you would say you keep the same phenotypes in the same cage. And in that case, you have eToVision tracking multiple animals at the same time in the cage. But eventually what you will get is the grouped data. Since eToVision can't remember if one animal crawls over another or one goes into the shelter and another one walks out, you don't know, eToVision doesn't know what animal it was. So yes, you can keep multiple animals in it. Take into consideration that the data that you will gather is grouped data.
Okay. Then I have a question about the number of cages. That's an important financial issue. What is the usual group size? Is variability lower than in acute tests?
Yeah, so the number of cages is a financial issue. Yes, that's understanding. The nice thing about the phenotypers is that the initial cost of what you have is the software and the first phenotypers with the camera overlay. Details I won't bother you with. But the software is the initial cost, and then by extending the amount of cages, it isn't so that if you buy the eToVision with 4 cages, that if you go to 8 cages, the cost double. No, not at all. It flattens out. The curve flattens a little bit, especially with the amount of cages that are added. So what is the usual group size? It depends. So we see that pharma companies, they do high throughput testing. They have 16 or even 32 cages active at the same time. Universities, it's mostly variable between 4 and 16 cages. However, more is sometimes also happening. And then you also mentioned, is the variability lower than in a Q-test? What I found, yes. So I found a high variability in grooming and rearing behavior, for instance, in these stereotypical autism mice. And this variance was way lower over longer tests. But again, take into consideration, do you want to find acute effects? Test acutely. Do you want to find long-term effects? Test long-term. But the variability really went down in this case.
Okay, thank you. The next question is, what experience do you have with characterizing compounds in the phenotyper?
Good experience. Yeah. So a very simple test that I did, because when I came there at Hoffmann La Roche, I was the first one to really work with them. So I needed to validate all the tools that I was working with. So characterizing compounds, amphetamine, that's a very known compound, I think. And characterizing it in the phenotype was really interesting, especially the data from that, because we saw we are all familiar with the acute effects of amphetamine, and that the mice in different doses, for instance, start making a lot of circles, circle not being very active. We also see it in fruit flies. So actually, characterizing the compounds and the effects that they have was very important to do. In many cases, however, I was kind of blinded. You need to be blinded in essence to make sure that you know what you're doing. But no, the character of the compound is quite visible. Yeah.
Okay. Then I have a question about long-term experiments. I'll just read it. It says, I'm performing hanging wire tests, and I've noticed that the difference visible at a short time point disappears in a long-term experiment. And maybe that's due to habituation. I'm not sure how to interpret these observations.
Yeah, that's, well, and first of all, I want to say you are the expert in this. You are the expert as a researcher to make an indication about these observations. But from my own experience, let's compare it to the grooming that I saw in the short term and I didn't see in the long term. Does that mean that my data on the short term is then worthless? No, it's not. It means that there is a certain factor that makes sure that that behavior happens. And in my case, it was the handling and the acute environment change and the stress. And maybe you can compare it a little bit with the stress and the environment changes in autistic humans. Then you will find these effects. So effects due to stress, effects due to acute handling, but you don't find them in the long term. With the hanging wire test that they habituate to what they are doing, yeah, my personal opinion, I would think the same. I would also think this is probably due to habituation, just how to control. Look at the Porsche for swimming test. If you put in your animals long enough, they will figure out that they are able to float. And if they're floating, at the beginning you would say that is due to them giving up. That was it. But in the long term, you would say they actually figured it out. They don't need to be so active. They can float.
Back to the compounds in Phenotyper. Do you know whether anything is published on this subject?
I don't know specifically what compounds you mean, but there's many publications on the phenotypes. I did a quick search, and yeah, you have a little bit stained with mentioning of references, but there's over 12,000 publications that at least have it mentioned somewhere. But there's only over 1,500 publications that have the phenotyper mentioned in their research. So I'm sure. And if you really want to know, you can contact, for instance, your local account manager. You can contact Noldus via the info website, and we will help you with it and actually refer you to these researchers, to this research that has been conducted. Yeah.
Okay, very good. Then I have a question about tracking multiple animals in multiple environments in etovision. So the question is, does etovision allow tracking of multiple animals in multiple environments, for instance, 2 or more separate arenas at the same time?
Yeah, again, so this is a little bit of an extension. So we know that it is possible. Let's see if I have a nice picture for you on this. To give you a good overview of that. I think going too much throughout the slides. Let's just look at this depiction here. So this is a graphical representation of what it looks like. You have 4 arenas. And with the phenotyper, this also means you have 4 arenas visible in the screen, and etovision is analyzing them at the same time. If you have 16 phenotypers connected, you have 16 phenotypers in view analyzed at the same time. And now your question is an extension to that. Is it possible to analyze multiple animals in multiple arenas at the same time? Short answer, yes, definitely possible. Take into consideration the computer power necessary to do these calculations, but this is something where we as Noldus will help you with. We will make sure that it is possible to do that, but having 2 separate arenas at the same time with multiple animals, no problem at all.
Okay, thank you. For the moment, these are all the questions I have, but let's wait a little bit, maybe more will come up. But if not, then we'll end this webinar. I don't see any coming up anymore. Another one. No, just a thank you. Okay, Romain, I think we ended here. If you have any more questions, you can always contact us. Thank you very much for attending. Thank you, Romain, for the interesting talk, and have a good day and see you again.
Thank you for your attention. Pleasure to be of help.